Well, hello everyone, and welcome to yet another episode of Drugbank in Conversation. Today, we have the great pleasure of being joined by Dr. Bousman. My name is Chris, and I'm the scientific support lead here at Drugbank. I'll be the host and moderator of today's discussion. So today, we'll be hearing about the 5 W's of pharmacogenetic testing, so specifically the who, what, when, where, and why of pharmacogenetic or genomic testing in pediatric and adult clinical settings based on current scientific evidence and real-world implementation experiences. And I think I speak for everyone when I say that we're all very excited for today's presentation and discussion. I think it's going to be a really interesting topic. So first of all, I'll just introduce our speaker, and then we'll go over the presentation and have a bit of time at the end for Q&A and wrap up. So a bit about our speaker today. So Dr. Bousman is an associate professor in the Department of Medical Genetics at the University of Calgary, where he leads the Psychiatric Pharmacogenomics Laboratory. This program of research spans pharmacogenomic discovery and clinical implementation, with particular attention given to improving prescribing for mental health conditions across life stages. He has published over 150 peer-reviewed papers and has received research support from numerous Canadian, US, and Australian funding bodies. He's also a member of the American College of Neuropsychopharmacology, the Clinical Pharmacogenetics Implementation Consortium, or CPIC, the Pharmacogene Variation Consortium, or PharmVar, the Pharmacogenomics Global Research Network, and the Standardized Laboratory Practices in Pharmacogenomics, or STRIPE Collaborative. Finally, he is also the founder and CEO of Sequence to Script Inc., a software company that assists laboratories with translating pharmacogenomic information into clinical action. So I think the background really speaks for itself, and I'm very excited to hear your presentation. So with that, I will pass things over to Dr. Bousman, and we can get started.
All right. All right, well, thank you for that kind introduction. So today, the main goal here is to answer, as my title said, the 5 whys of pharmacogenetics. So why, what, who, where, and when. And so I'm going to go through each of these in a sequential order. I'm going to start with why. So I like to start with this quote for all of my talks, because I think it really captures the current state of prescribing, but I also think it really highlights the need for more personalized prescribing approaches. And if we think about this in a different way, we know at the population level, medications are efficacious and tolerable. If they weren't, they wouldn't be approved by regulatory bodies such as the FDA or Health Canada. But if I take someone randomly out of the population and look at the individual level, we know that efficacy and tolerability can vary quite substantially. And so we're left with this challenge of determining which drug fits in which one of these quadrants for a particular individual. And again, really highlighting the need for more personalized prescribing strategies. So here I'm showing a few of the personalized prescribing strategies that already exist and are being used. Most of these are probably familiar to the audience. They've been around for decades and are used as part of standard of care. Of course, today we're going to focus on pharmacogenetics as a new strategy to complement or enhance these other strategies. And I want to emphasize that pharmacogenetics is not meant to replace any of these strategies. So what is pharmacogenetics? So basically, it's the use of genetic information to predict a person's ability to process and react to medications. We typically lump this genetic information into 2 broad categories. So we have the pharmacodynamics. So these are genes that are associated with what a drug does to the body, or the receptors, ion channels, signaling pathways. And then we have the genetic information associated with pharmacokinetics, or what the body does to a drug. So these are the genes or enzymes affiliated with absorption, distribution, metabolism, and elimination of drugs. And it's this category that has garnered the most attention in pharmacogenomics and where the most of the evidence and implementation is occurring. And more specifically, the metabolism area within pharmacokinetics. So when we think of metabolism with a pharmacogenetic lens, we typically will think of drug metabolizing enzymes. And now, fortunately, over the last decades, a number of pharmacokinetic studies have done a good job of linking genetic information or genotype in these drug metabolizing enzymes to the metabolic ratio, where we can then make pretty reasonable predictions about a person's metabolizer phenotype. And this typically goes from poor on one side, or slow metabolism or enzyme activity, to the other side of the spectrum of ultra rapid or fast enzyme activity. And if we know someone's predicted phenotype, we can usually do a reasonable job of predicting how much of a drug will be in their blood at a standard dose. So those on that poor or slow side of the spectrum are going to be at greater risk of having a higher exposure to the medication and be at higher risk for side effects. Whereas those on the other side of the spectrum, that ultra rapid, are going to be at a higher risk of receiving no benefit at the standard dose because they're not going to get enough concentration or exposure to the drug. So the question always is, what genes and drugs can we apply this prediction to, or this pharmacogenetic implications? And so, of course, in order to figure that out, we need to go back to the evidence. And in pharmacogenomics, that's both the published literature as well as the drug labels. Now, fortunately, we have a group known as the Pharmacogenomics Knowledge Base, or PharmGKB, that pulls out all of the gene-drug pair associations from the published literature and the drug labels, and then curates these and ranks them on a strength of evidence scale, ranging from unsupported to high. So the unsupported, as the label would suggest, is gene-drug pairs that there's really no evidence to support an association. The low level of evidence is where we start seeing single positive studies for a particular gene-drug pair, or there might be a number of studies, but there's conflicting findings. Moderate level is where we start seeing multiple replications, but the effect size is small, and some would argue maybe not relevant to clinical practice or not ready to go into clinical practice. And then the high level of evidence is where we see multiple replications of large effect, and this is where we say this is ready for the clinic. And it's these high-level gene-drug pairs that get pushed forward into the prescribing guidelines development pipeline. And here I'm just showing you the four main guideline developer groups, the top two being the most prominent in the field, the top one being Clinical Pharmacogenomics Implementation Consortium, or CPIC, which is based in the US but has an international membership. And then the Dutch Pharmacogenomics Working Group, which is obviously in the Netherlands. And there's groups here in Canada as well as in France that create guidelines as well. Altogether, these groups have created guidelines for 87 medications. And I'm showing those here in this busy network plot. And it's not easy to see everything, the drugs and the genes, but the reason I'm putting this up here is really to show you that pharmacogenomics reaches a wide scope of medicine. It's not really specific to any one area of medicine. It really does reach a number of areas. So we have these guidelines, but who can benefit from pharmacogenetic testing? So I want to start by answering this question with a case example of a 23-year-old male, and he presented with repetitive and obsessive behaviors. He had anxiety related to visual and auditory stimuli, and he had difficulties with transitions. And he was prescribed fluvoxamine, which is an SSRI. And on this medication, he began to exhibit regressive behaviors, and as a result, his part-time employment was terminated, and he was required to return to his family home. Upon his return, his clinical team did a full clinical workup, so brain scans, CSF analysis, muscle biopsies, full blood panels, but they really didn't find anything outside the normal range. And it was at this stage that the patient's psychiatrist recommended pharmacogenetic testing. So here I'm showing you a summary of that pharmacogenetic testing results. And there's a lot to unpack here, but I'm going to just bring your attention to the CYP2D6 result, where it shows that he was predicted to be a poor metabolizer. So as I recall, he was prescribed fluvoxamine, and fluvoxamine is primarily metabolized by CYP2D6 into inactive metabolites that are then cleared from the body. But because this gentleman is a CYP2D6 poor metabolizer, he's unable to break fluvoxamine down at its regular rate and clear that clear the drug from the body, which puts him at risk of overexposure, putting him in that toxic zone, and putting him at risk for side effects. Now, this is a relatively well-known interaction, and this is what led to CPIC recommending that individuals that are CYP2D6 poor metabolizers, we should consider 25 to 50% dose reduction, or potentially select an alternative drug that's not metabolized by CYP2D6. And the FDA more or less concurs, as suggesting that if you're a poor metabolizer, CYP2D6 poor metabolizer, that you're going to have higher systemic concentrations, and that we should use fluvoxamine with caution. So based on this information, the treatment team decided to discontinue fluvoxamine, and they commenced desvenlafaxine, which is another antidepressant, but it's not metabolized by CYP2D6. And he had progressive improvements in all behaviors and sleep, and he returned to his supportive living home and commenced work again. So the take-home message here for this case is that pharmacogenetic testing could really significantly reduce these types of experiences, and also importantly, avoid unnecessary medical procedures. If you recall, when he returned to his clinical team, they did that full workup, including the CSF and brain scans, so quite expensive and invasive procedures that may not have been needed if the pharmacogenetic testing was available sooner. So that's an example at the N of 1 level, but there's been a number of studies published, and this is just a subsample of those, showing that pharmacogenetic testing is efficacious not only in psychiatry, but cardiology, cancer, neurology, treating pain, as well as gastroenterology. And just to give it a bit more granularity, I'm going to break it down by 3 broad age groups. So if we start with the young people, this is where the evidence is still emerging, but arguably the ideal population to implement pharmacogenetic testing. And it's because I see it as the greatest cost savings potential. I mean, these young people have their whole lives ahead of them. Having the pharmacogenetic testing available now, they can carry that through their life, and it can help with prescribing throughout their life. The most common indications to date, and this is obviously not exhaustive, but just highlighting a few of the low-hanging fruit. Psychiatry definitely being that, with antidepressants and antipsychotics. I'm purposely putting stimulants here with that thumbs down because this is where we don't have a lot of evidence, but we need more research. And just to be cautious when ordering testing, if stimulants is what you're looking for advice on, we really don't have it at this point in time. Oncology as well as gastroenterology are also users of pharmacogenetic testing within the young people population. When we go into adults, this is where the strongest evidence lies and where most of the implementation is occurring. The clinical indications here is basically anyone starting a drug with a PGX guideline. So one of those 87 drugs that I shown in that figure earlier. Individuals that are not showing any response despite high adherence or having side effects at very low doses would also be great candidates for pharmacogenetic testing. And there's some emerging evidence to suggest if someone is reluctant to start pharmacotherapy or has low adherence to pharmacotherapy, that pharmacogenetic testing may be an aid in that. And the indications are very broad, of course, but again, psychiatry, pain management, cardiology, and oncology tend to be the ones that are doing the most uptake for pharmacogenetic testing. And then finally, the older adults, similar to young people, the evidence is still emerging, but this is arguably the most challenging population to implement testing. And this has to do with higher frequency of polypharmacy in this age group, as well as well-known and documented pharmacokinetic changes that occur as we age. The clinical indications are more or less the same as adults. There is some emerging evidence to suggest pharmacogenetic testing may help with deprescribing decision-making. So that's something to keep an eye on as well. Okay, so one of the biggest questions I often get asked is, where can pharmacogenetic testing be ordered? There's hundreds of labs, if not approaching 1,000, around the world that will offer pharmacogenetic testing. So how do we determine which lab we should order from? Because me and my colleagues get asked this question so often, we did develop this decision tree and guide to test selection. It's probably too small on your screen to actually look at, but I'm just going to boil it down into its 4 main best practices. So when you're choosing a testing lab, excuse me, the first best practice is it needs to be evidence-based. So we want to make sure the lab is following the guidelines. So the CPIC, Dutch, FDA, the guidelines that exist. Number 2, we want to make sure that the lab is accredited. So CAP, CLIA, or some kind of equivalent accreditation to ensure a high standard of analytical validity. We also want them to be transparent. And so what I mean by that is they need to be clear about what and how they're testing, but also, and probably more importantly, how are they translating the results into recommendations? And if the lab isn't able to tell you this, this is where, if not able to or doesn't want to, then this is where you'd want to look elsewhere. And then finally, the lab should be supportive. And what I mean by that is provide some type of interpretive support, either through some web portal or access to a consult through pharmacist, or someone that has a background in pharmacogenomics if needed. And the last thing I'll say about this is of all the labs that I've been able to evaluate, it's clear that there is no superior lab to all others, but there are inferior labs. And in order to avoid those inferior labs, you just need to follow these 4 best practices. Okay, so one of the issues when you're selecting a lab as well is that we need to really be aware of potential cautionary content. And this, what I mean by cautionary content is, or why cautionary content comes up is regulatory standards for test content have not been established. So there are no rules about what a lab can and cannot test or what genes they can put on their panel. And this leads to content that has good face validity, good biological plausibility, but inconclusive clinical validity. And so I'm providing here a few examples of what I mean by these cautionary gene-drug pairs. And these are all examples from psychiatry, but they exist in all areas of pharmacogenomics. And so you can see these 5 examples. They all really have good face validity and biological plausibility. But if you look into the literature and the data supporting the use of these gene-drug pairs in practice, it's really quite inconclusive and not really ready to be implemented. And to give you an idea of how often these gene-drug pairs occur in laboratory tests, you can see it's a non-trivial amount. And this is why this needs to be on our radar when interpreting tests. I'm certainly not saying that you shouldn't order a test from a lab that includes one of these gene-drug pairs because it might reduce the number of labs substantially. But if they do include some of these, you probably want to be very cautious about how you implement them. And my advice would be to ignore the results for any of these gene-drug pairs. And the reason why I advise that is based on this second case that I want to present to you. And this is a 25-year-old male with schizophrenia, and he was admitted to hospital on an involuntary basis due to bizarre behavior and command hallucinations. He had a history of multiple medication failures, and this is what led the treatment team to decide clozapine was the next best step. So the problem was the patient's family and family doctor opposed this decision based on pharmacogenetic testing results that were ordered by the patient's family doctor. And so here's the results of that, the summary of those results. And you can see for the most part, there's not much to be said. He's a normal metabolizer across the board. But this particular lab that they ordered the testing from included a test or gene DRD2, so the D2 dopamine receptor. And they said that this gentleman's genotype would predict him to be a poor responder, and they specifically highlight clozapine as one of those drugs he would be a poor responder to. So based on these results, the patient's family and family doctors said, no, we don't want him to be started on clozapine. So let's unpack this a bit and look at the evidence for DRD2 and clozapine. So first, if we look at the drug labels, all of the major drug labels have no mention of DRD2 genetic variation have any effect on clozapine response. There are no dosing guidelines associated with clozapine or DRD2 at the current among any of the primary guideline developers. And if we look at PharmGKB and how they rank this gene-drug pair and the strength of evidence scale, they currently have it as a low strength of evidence, meaning, again, not yet replicated or conflicting findings. And in this case, it's the latter. So based on this information, clozapine was commenced after getting approval from the medical director and the patient consented. And the patient responded rapidly and was discharged. And he gradually returned to his previous level of functioning. So the take-home message here is that we really need to be cognizant of only implementing gene-drug pairs that have prescribing guidelines. When we stray away from the guidelines, we do run the risk of potentially taking efficacious medications off the table, or the other side is prescribing medications that are potentially inappropriate for a particular patient. Okay, so we now know a bit about where, who, and why, but how about when? When should we be ordering pharmacogenetic testing? And this is really an open question, but there are three, I guess, broad ways that you can look at this. So the first is a reactive testing approach, and this is the most common approach currently. And this is where we're offering pharmacogenetic testing after someone has already exhibited a poor or experienced a poor response or side effects to a medication. And the reason why it's the most common is a couple of things. One is the reimbursement kind of dictates a reactive approach currently. And so it's also kind of a cost savings measure for the payers, in that we don't want to test everyone. We want to wait and see who really could potentially benefit from this. So unfortunately, patients must have a bad experience before they can get pharmacogenetic testing. There's also the point of care approach, where pharmacogenetic testing is offered at the same time the initial drug selection and dosing decisions are being made. And then the notion being that the results will be ready within about 2 weeks, and that adjustments could be made early on in the treatment process versus waiting later until there might be a bad reaction or a poor response. And then the final is the preemptive, which most of us in the field would suggest is the ideal, offering pharmacogenetic testing prior to making drug selection and dosing decisions. And this is where we're hoping to move towards in the field in the future. Okay, so those are the 5 W's, but I'd like to offer a bonus. It doesn't start with a W, so look more about the how, and just briefly touch on how to integrate pharmacogenetics into practice. How do we interpret this data? And always a big question is, how much does it cost? So first, how do we integrate? So ideally, if we really wanted to have the largest impact the pharmacogenetic test results need to be integrated into electronic medical and health records. Now, few institutions globally have accomplished this. And I'd say the majority of pharmacogenetic implementation programs are in this level 4 or level 3, which is where we have standalone reporting, PDF kind of static reports that may or may not be uploaded into the EHR, but they may be in some obscure folder where the ordering physician knows where to find it, but no other physician down the line that the patient might encounter will know where to find the information and may never know that they were tested. So what we want to do is obviously shift towards this level 2 or level 1, where it becomes more fully automated and integrated into the EHR, where we have alerts that can be triggered, letting physicians know when they're prescribing a medication that there is an actionable result, and it can help with that clinical decision support. We also know that when we're interpreting results, it does matter which lab it comes from. Recommendations do vary from lab to lab. This is something that we found when we did this small head-to-head comparison, where we recruited 5 patients with treatment-resistant depression. And we asked them to give us, each patient to give us 4 samples of saliva. And then we sent each sample to 4 different labs. And we picked 4 labs at that time that the only 4 labs that had randomized controlled trial evidence supporting their test. That's the rationale for those 4. And then when the results came back, we compared the genotype and phenotype agreement as well as the medication recommendation agreements. Now, what we found was when we compared genotype and phenotype, there was pretty good concordance. So no matter which lab we looked at for a particular patient, their genotypes and phenotypes were had high concordance, I'd say above 85%. But when we looked at the recommendations, like what recommendations they were providing, that's where we've seen less than 60% agreement in the advice across the 4 tests or 4 labs. And this is didn't really matter which indication or each medication class we were looking at, we see this poor agreement. And the take home here, of course, is that we can't assume that a lab's test results are equivalent or interchangeable to another lab. But this also raised the question of why. Why are we getting good genotype and phenotype agreement, but then the medication recommendations are not agreeing at all? And the answer partially is many of these labs take a black box approach to interpreting the information. So as I mentioned, they have good agreement on the genotype, phenotype, but how they take that information and translate it into medication dosing and selection recommendations is sometimes unknown. They're using decision algorithms that may be proprietary. We do typically know the ingredients within that algorithm. So they often will say they use the published literature, drug labels, they may mention PharmGKB, and they might mention some of the guidelines. But we often don't know how they're weighting that information. And this is where we think that that's why we're getting this discordance from the genotype to the phenotype and then the recommendations. So what do we do about this? Well, one thing we can do is bypass the black box. As I mentioned, we're pretty confident with the genotype results and even the phenotype results for most of the labs. So that seems to be consistent across labs. So we can take that information directly into the guidelines. The guidelines are freely available, publicly available. And so you can go and cross-reference a patient's genetic information directly to the guidelines. Now, of course, busy clinicians will wave their hands when I say this, because who has the time to do that for each one of their patients with a report in hand? And we agree. And this is one of the reasons why we built a second opinion tool known as Sequence to Script. And so basically this allows physicians, pharmacists, whoever's receiving pharmacogenetic test results to plug that information into Sequence to Script, and it will generate a report that's strictly based off the guidelines. And so it truly acts as a second opinion in determining whether or not the lab that was doing the testing for this particular patient is following the guidelines, and how much you want to follow that report in the future. And then the final thing around interpretation that I want to mention is that, and it's often overlooked, is that non-genetic factors can play an important impact on interpretation. And this is due to a phenomenon known as phenoconversion. And this is where we see a mismatch between a person's genotype predicted phenotype and their clinically observed phenotype. And so just to give you an example, let's say that I'm going to genotype someone for CYP2D6, and the results come back from the lab and they're a normal metabolizer. So that's great. Normal metabolizer, typically we're going to just use standard dosing procedures. But before we do that, we want to look at their current medication list, and we notice, oh, this person is currently taking fluoxetine. Now we know fluoxetine is a potent CYP2D6 inhibitor. So while this patient is taking fluoxetine, although their genotype, predicted phenotype is normal, because of this inhibitor is on board, they will phenoconvert to a poor metabolizer as long as they're taking fluoxetine. And so this is something that we definitely need to take into account, and it's something that occurs quite frequently. And I'm showing here a study that was done about a decade ago, but still very relevant, of 900 patients that were treated for depression. And what they did is they genotyped all these 900 first, and they seen for CYP2D6, 4% were poor metabolizers. And that's basically what we'd expect in a general or in this population. This is a European, primarily European background population. So we'd expect around 4% to be poor metabolizers. But then they looked at their current medication list of all these 900 patients, and based on that, they determined 27% would actually be poor metabolizer phenocopies, or phenoconverted to poor metabolizers. So this is just showing that we have to take into account these non-genetic factors, in this case, concomitant medications, when interpreting pharmacogenetic test results. And just as some information for you, there are free phenoconversion tools. I've already mentioned Sequence to Script, which does account for phenoconversion, and there's also one out of the University of Florida that does it specifically for CYP2D6, if that's the only gene you're interested in. Okay, and then the final thing I'm going to touch on is always the elephant in the room. How much does this testing cost? Well, there's a wide range of prices, and it really depends where in the world you're doing the testing. But more or less, what I've been able to kind of collect is it's going to be somewhere around 150 to 2,000 plus US dollars. And we know that reimbursement is variable and inequitable, depending on where we're doing the testing. And the other thing to keep in mind is the test cost does not correlate with the quality of the test. So we've actually done this correlation based on some of our evaluations of how strictly labs follow the CPIC guidelines. And definitely the most expensive tests don't necessarily follow the guidelines the best. So you don't base the quality of a test on the test cost. And then related to cost is obviously cost effectiveness and cost savings. And I can say this was a study that was just done last year, a systematic review of 108 economic evaluations of pharmacogenetic testing. And they found that for the most part, you can see the two green slices of the pie there. Most pharmacogenetic testing is cost effective or a cost saving strategy when compared to standard of care. So there certainly is a case to be made that not only does it have a clinical impact, clinical efficacy, but a cost effectiveness as well. Okay, so I'll wrap up with some 3 take-home points. So first, I hope I've made it clear that pharmacogenetic testing is an evidence-based strategy to inform prescribing of medications across a person's lifespan. Also, that the impact of pharmacogenetics really does depend heavily on the quality of the testing and the appropriate interpretation of results, specifically when I was just talking about those non-genetic factors. And then finally, just to reiterate what I said up at the top of the presentation, that pharmacogenetic testing is a companion decision support tool that can enhance, not replace, current prescribing strategies. All right, I'll stop there, and happy to answer any questions anyone has. Thank you.
Yeah, there we go. I'm waiting for my camera to return. Yeah, no, excellent. Thank you so much for that wonderful presentation. I think that may have been the clearest and most concise overview of pharmacogenic testing that I've ever seen. So very, very useful, I think, for everyone watching this now and for watching the recorded version later. So we're now opening it up to Q&A. So feel free to pop your questions in the chat bar or use the question function. And while we're waiting for those to come in, I think if it's all right, I have a few questions that I'd like to ask given this opportunity as well. So the first one is sort of around, and I'm not going to pretend to be an expert in this field, but I've noticed that there are a variety of different ways of referring to genetic variants associated with different outcomes to medication. So there's things like the star nomenclature. There's a different nomenclature that's used for HLA and HLB. And then, of course, you can always just call the specific variant on a reference sequence. And I'm wondering to what extent those different ways of representing what is essentially the same information can have impacts on sort of the recording and interpretation of variants.
Yeah, I mean, there's been big attempts to get a consensus nomenclature. It's definitely been done for CYP2D6, which is was very helpful in defining the star nomenclature. And there's a group known as PharmVar. You mentioned in your introduction to me that I'm a member of PharmVar, but that's their main goal, is to make sure that we're all using the same language when we're calling these variants, because there can be confusion, right? And where there's confusion, there's room for errors. And so, yeah, the nomenclature does matter. It might not be the sexiest thing to do, but it's a definite necessity. It's like a foundational thing for pharmacogenetics to thrive. Is we have to all make sure we're talking the same language. Yeah, so PharmVar is probably the leader there in making sure that we're all on the same page. HLA is kind of in its own world, I guess, but they have stars as well. They used kind of a star, but it's their own kind of way of doing it. But yeah, the goal is to, I think, use a star or adopt that star nomenclature for all pharmacogenes. I think that's kind of the way of future. There are still some, obviously, pharmacogenomic genes that do not have star allele nomenclature, and I would argue those are the most difficult to implement and to discuss with colleagues and to report in publications, because you have to do a lot of legwork to, are they looking at the same thing we're looking at? In which build are they using? Which genome build are they using? So it can be challenging. And PharmVar kind of brings that all in under one roof and calls an apple and an orange, and it makes things a lot easier.
Yeah, that's comforting to know. I think that's always been one aspect of the PGX landscape that I've personally found confusing, and I think probably other people find it confusing too, because if you see all these different things popping up all over the place, it can be difficult to know where to ground yourself. I'm just going to check really quickly because there's a message in the chat. Okay, so we have a question here. So this person asks, why do you think that the adoption is still lagging at this point, given that it is, as you said, both sort of cost effective and has clinical benefit?
Yeah, I mean, there's many ways to answer this question. I'll go with my bias, where I think one of the main reasons is we have obviously the CPIC and Dutch guidelines, but we don't have clinical practice guidelines that are adopting those prescribing guidelines. So doctors tend to read clinical practice guidelines, not the CPIC and Dutch guidelines, right? So some may be aware of it, but most are not. They look at their society, whatever society they're a member of. So the American Heart Association, if you're a cardiologist, the American Psychiatric Association, if you're a psychiatrist, or whatever country you're in that society. And if there's no guidelines saying, yes, pharmacogenetic testing should be considered, then it's kind of, they don't think about it. They don't think it's time to go. So as a pharmacogenetics community, we're starting to work and trying to focus more on those committees that are making those guidelines and doing some education with them, and letting them know that where we are at in the science, because we're way advanced in many of these areas where, to me, it's almost malpractice not to offer pharmacogenetic testing. But sometimes the guidelines aren't just there. We do know that in general, this isn't just for pharmacogenomics, but it often takes 10 years to get good science into practice guidelines. It's just, there's a lag. And so I think it's happening. In this case, if you live in the US, you know reimbursement's happening already. So insurers and people that are paying for healthcare are seeing the benefit before the clinical guidelines are seeing it. So I think that's a little bit of showing how it's moving. We have insurers that are willing to pay and reimburse pharmacogenetic testing. And I think it's just a matter of time before the clinical societies get on board. But they're inherently conservative. They always have been. So I think that's just, we have to work with that.
Yeah. Well, perhaps being overly conservative in matters of healthcare isn't always such a bad thing.
No.
Yeah, I could see being cautious as a benefit. I want maybe just to follow up on that question, because I was going to ask if it hadn't come up about the reimbursement side of things, and I'm thinking specifically in places like Canada where there is a public health system. To what extent is that kind of a hill to climb in terms of convincing maybe a body that doesn't have as much leeway as a private insurance company that they should reimburse these kinds of tests?
Yeah, so I think in Canada, I can't speak for all of Canada, but at least in Alberta, where I'm situated, I think this is where the cost effectiveness studies and modeling specific to the province, or maybe all of Canada, but I think these are province by province decisions, right? So every province is going to have their own threshold, but I can say in Alberta, it's very much a question of, if we offer this testing, not almost how much is it going to cost our system, but how much are we going to save or divert? And without really solid numbers and modeling, it's really uphill battle, right? Most of the people I talk in the healthcare system, the healthcare administrators, they don't need convincing that pharmacogenetic testing is efficacious and important, but that's not enough. They need to know that it's not going to break the bank in a public healthcare system. Why should we cover pharmacogenetic testing over another innovation? Like, give us the ammo we need to drive this. So I think, and that's probably the same across all public healthcare systems around the world that have that same socialized kind of medicine. In the US, obviously, it's driven by other forces, and I think that's why they're first out of the gate in this case. But I will say what we're seeing in the US is definitely kickstarting conversations in places like Canada, because they're like, oh, because a lot of people are looking to the US to see how is this is like an experiment. Let's see how this works. And I do think it is going to happen in Canada. We're just waiting for really local good data to show us that this isn't going to be a burden on our system. This is actually going to reduce burden.
Yeah, of course, that makes sense. So we have another question from the audience here. And this individual says, based on recent developments with the FDA rejecting a CDS's application as a medical device, do you have any comments on utilizing FDA versus CPIC versus other guidelines?
Yeah, this is a hot question right now, isn't it? So I think everybody has an opinion here about what the FDA is doing and whether whether there's overreach or that it's in their rights. I'm not going to take a position here, but what I do know is that it's making a lot of uncertainty in the area, which is obviously not helpful to anyone. Our patients being the number one. So yeah, it's hard to answer that question. What I will say is that I think there's different solutions out there, and I can see that the person that answered or asked the question has one of the solutions. So maybe that's why they're interested. But I think it's going to be a case by case basis, and I think the FDA would like us all to just be following FDA drug labels. And I'd be happy to do that if they were updating them more frequently and they were up to date on the science. When a drug is sent and submitted and approved, that it's updated then, but how often does it get updated after that, the post-marketing kind of research? And the answer is not very often, unless something really bad happens and black box warnings appear. So I think that's where we're struggling. Like, well, yeah, we agree. FDA is great. We can follow them, and most of us do, but it needs to be supplemented with more up-to-date information that comes from like CPIC and the Dutch Pharmacogenomics work group. So this is a really challenging topic to discuss, particularly when you're talking to a government agency that doesn't really want to hear from us. It doesn't seem. We're going to do what we're going to do, live with it. So yeah, I don't have a really great answer for that, except for I feel the frustration in the field, and we're all kind of feels like we're walking on eggshells a bit at the moment.
Yeah, I think there's been a couple of FDA decisions as of late that have caused a little bit of controversy, but that's
definitely.
It's a discussion for another webinar, perhaps.
To do a whole talk on the regulation bit, not something probably, maybe for another day.
Okay, so the questions are coming in now. Which is great. So we have another person here saying, seems that public payers are approaching coverage on a single drug gene basis. Do you think that we may eventually see openness to considering coverage of multi-gene, multi-drug panels from a public perspective?
Yeah, I mean, this is in the US, it is happening. So if I'm not mistaken, Medicare will pay for up to a panel of 6 at a time. And United Healthcare, I think it's 12. Don't quote me on that, but it's in that neighborhood. So again, that's in the US. Very much could be in other places. It's a single gene by gene, which is obviously not helpful because that actually ends up costing the healthcare system more because you have to test. You're paying for an individual test where we know genotyping 12 genes is almost the same cost of genotyping 1. Very little difference because the way that the assays are set up. So yeah, to me, it's a no-brainer to do panel testing. Then you could truly, the bigger your panel, you can truly say this is a lifetime test. It's obviously not a lifetime test if you're only testing one gene drug pair. It's only a lifetime test for that particular gene, but not for pharmacogenomics in general.
Yeah, it's funny. We've really reached that point, I think, with a lot of different sequencing-based approaches where there is economy at scale. You can basically pay the same for, a couple of genes as you can for even a whole genome in some cases, depending on what you're talking about.
Yeah.
So I think if it's all right, I think if we can just do maybe 1 or 2 more questions and then we'll wrap up. I was curious because you were talking about that phenomenon of phenoconversion, and you gave the example of a drug interaction as a very real example where that kind of thing can happen. Are there other things besides just co-prescribed medications that can cause this phenoconversion phenomenon to occur?
Yeah, there's actually been some really nice reviews done in the last couple of years that not only show the concomitant medication, but lifestyle, environmental kind of stuff. So like smoking is a well-established one with CYP1A2. Now, CYP1A2 doesn't have any current guidelines associated with it, but it's always the poster child for phenoconversion because we know smoking predictably induces CYP1A2. And then there's pregnancy. Pregnancy can cause phenoconversion in some genes, CYP26, estrogen levels. Diet, there is dietary ones as well. Grapefruit juice is a big one with the CYP3A locus. Of course, people know about that one. So there's definitely other factors. And then just in general, like I mentioned when I was talking about the older age group, we know there's pharmacokinetic changes that exist. Our livers and kidneys don't continue to function at the highest level throughout our whole lives. They do start to wind down. And so we have to keep that in mind when a normal metabolizer, an 18-year-old, and a normal metabolizer of an 89-year-old could be different, even if they're not taking any other medications, just because of that aging effect. So yeah, I think there's a lot of phenoconversion. We literally are scratching the surface, I believe, and we tend to focus on the drugs, the medications. But I think we have a lot more to learn about how our environments affect not only how we process or metabolize, but even how some of the pharmacodynamic genes may be being affected, which is a whole other area that we are not really getting into in pharmacogenetics as much as we are with the pharmacokinetics.
Okay, perfect. Yeah, that was a great answer. Okay, so I think with that, we'll probably wrap up. We'll end a little bit early today, but that's fine. So I'd like to thank everybody for attending, either live or for signing up to watch the recording later. Of course, this webinar has been recorded and will be shared with all attendees and registrants via email in the next few days. If you're interested in learning more from Dr. Bousman, either about his research or about sequence to script, there's, of course, websites from the University of Calgary, from Sequence to Script itself. He also has a profile on LinkedIn, which you can find him at. And if you're interested in learning more about Drugbank, you can visit drugbank.com, follow us on social, do all of those great things. We'll have more fantastic webinars just like this one coming up in the future. And then finally, I'd just like to once again thank Dr. Bousman for his time and for the really great presentation and the discussion afterwards. We're very lucky to have had you.
Thank you.
Okay, so with that, we'll say goodbye and hope to see everybody at a future event.